Hello, everyone. My name is Olivia Long, and I will be your moderator today. I am absolutely thrilled to welcome you to today's webinar, titled From AI Design to Validated Binder, Closing the Loop with Reversible BLI Biosensors. We have an incredible session planned for you today, filled with insightful discussions and valuable takeaways. Before we get started, I'd like to extend a very warm welcome to our esteemed guest speakers. Dr. Alexandra Nordlund, Product Manager at Cube Biotech, Dr. Ron Gill, Innovation Manager at Adaptyv Bio, and Dr. Benjamin Osborne, Product Manager of Applications at Gator Bio. Please feel free to submit any questions you may have during the webinar today. To do this, simply head to the engagement window to the right of the auditorium and click to open the Q&A window. So, without further delay, I'm delighted to hand over to Dr. Nordlund, Dr. Gill, and Dr. Osborne, and I would like to thank you for presenting to us today.
Okay, well, thank you all for coming. I'm really glad to be here to present about BLI in the AI era, as well as introduce our co-presenters today. Thank you for taking the time to learn about these things and to hear from us. We invite your questions at the end. As everyone watching this knows, the AI era in drug discovery and biopharmaceuticals is here. And in silico approaches are able to cut the number of candidates that would need to be screened from the thousands of yesteryear down to dozens today. That means that you're able to trim the fat and cut right to the candidates that matter, the ones that are more likely to work. And that is a revolution in the drug discovery space, in the biologicals space. And as you know, AI is constantly improving. That means that you can actually, with a single core GPU, you can generate a protein ligand pair as a 3D complex in 20 seconds. This means that this is going to democratize the ability to generate these possible candidates. And it means that as we continue to improve these processing capabilities, better and better models will become available, and undruggable candidates with various point mutations that used to be undruggable can now be targeted. But however, no matter how good your AI methodology or algorithms may be, you need to still make those proteins. And you need a way to capture them. You need a way to study them interacting with their targets. You need to observe their stability. You need real binding data. You need off-target screens. You need to confirm the epitope assignment. All these things still have to be done regardless of how good the in silico data may look. So AI-fueled wet labs need the ability to quickly process many, many samples, but maintain a high quality in the data that they generate. They also need a variety of ways to immobilize their candidates and targets, because the immobilization strategy can vary from project to project, depending on the protein or peptide being studied. You also need sensitivity. You need to be able to observe the interactions of very small peptides or nanobodies or similar constructs, because a variety of targets aren't these large proteins that we used to target, but a lot of them are much smaller. This also means that you need output from the data that you generate that can be fed back to the AI to train it and make it better at its job. This is where Gator Bio really excels in all of these ways. We're founded by industry veterans, and that means that we have the expertise that leads to quality at scale. We also are very innovation focused. We have a variety of immobilization methods, but one of our big innovations is making biosensors that are completely compatible with cell-free, which is the primary method for expressing proteins in the AI era. And we're a team of passionate experts, and we really only do biolayer interferometry, this one technique that's used for measuring all of these things. That means that we know how to help you. And so if you could reach out to us, contact us, we can provide you the support you need as you scale up your processes. One of our most fascinating innovations at Gator is our XT biosensors. Much of the light reflection from the protein layer As proteins build up onto our biosensors, much of the light is actually scattered and lost. Gator's XT biosensors collect more of that light, creating more signal per molecule. That's really helpful because it can triple your signal, as shown here on the right. It will increase the total amount of signal without increasing the total number of molecules, meaning you don't need to overload your sensor just to see smaller molecules binding. And this allows you to avoid issues with avidity and mass transport. Working with Gator, you also have access to a variety of biosensors that are completely unique. Our HIS-XT biosensor is an AI-designed single-domain capture for less avidity and higher sensitivity for capturing his-tagged proteins from cell-free and lysate. It's also fantastic for Fc receptors because it doesn't have an Fc domain. We also offer Strep-Tactin XT biosensors, which I think are one of my favorites, and you're going to hear more about them from Cube Bio here in a minute. These biosensors capture ligands with picomolar affinity, making them a top-tier choice for a variety of these AI workflows. We offer a variety of other biosensors as well, like anti-VHH and streptavidin, as well as many others, depending on your application needs. Now, I don't want to keep talking too much because I think you'll want to hear more from Cube and Adaptyv. Once again, thank you for your time. We look forward to hearing your questions at the end of the webinar.
Hi everyone, thank you for joining this webinar. In my presentation, I'm going to explain to you what the differences are between biotinylation versus Strep-Tag2 and Twin-Strep-tag, with a focus on which tag ligand combination is actually suitable for BLI experiments. But first, I want to say a few words about our company. Some of you might associate the Strep tag with IBA Life Sciences, but IBA Life Sciences was acquired by Cube Biotech at the beginning of last year, and now we're currently working on unifying our brands to Cube Biotech. However, if you now, after this webinar, search for Strep tag or Strep-Tactin, you will likely still end up on the IBA Life Sciences website. That is because we're currently still in the transitioning phase. Both companies are actually located in Germany, and we develop and manufacture the majority of our products in-house, and we also sell them worldwide. And especially for our US customers, we also have a distribution facility in the US to guarantee quick deliveries. The reason why I'm addressing this topic today is because we noticed this with our customers, but we also got the feedback from Gator that there's often confusion around the streptavidin, Strep-Tactin, and Strep-Tactin XT systems, especially what is what and what can I use with which ligand. So I hope I can clarify this for you today. And so after this webinar, you know exactly which tag ligand combination you can use for what application. I will start now with the origin of the Strep tag technology, which also will give you an idea why there's so much confusion around it, because the systems are really strongly connected to each other. So the Strep tag technology is actually based on the streptavidin biotin interaction, which is one of the strongest non-covalent interactions found in nature, which is of course great if you want to immobilize your protein. But it comes with limitations if you, for example, want to use it for purification, because it's really nearly irreversible. So the goal was to have really a highly specific system, but that is still reversible. So first, a peptide sequence was developed, the Strep tag, that binds into the biotin binding pockets of streptavidin. But the binding was not very strong, and therefore the binding pockets or the biotin binding pockets of streptavidin were mutated as well, and this resulted in Strep-Tactin. And this is basically the first system that worked really well for protein purification. To now get an even increased affinity, Strep-Tactin was mutated again, so again the biotin binding pockets, and this resulted in Strep-Tactin XT, which binds with a higher affinity to the Strep tag 2, but also to the Twin-Strep-tag. So the Twin-Strep-tag is basically just the strep-tag duplicated and connected with a linker. So it not only binds into one biotin binding pocket, so actually into two pockets. So you get an even stronger binding. So basically you get from streptavidin to Strep-Tactin XT an increase in affinity to the strep tags and at the same time a decreasing affinity to biotin. So if you now look at the systems, you get, of course, the highest affinity if you choose streptavidin in combination with biotin-related proteins, but you have the disadvantage that it's irreversible. And when you work with the strep-tag system, you have different affinities available with the advantage that it is reversible. So now that combination that actually offers the advantage of having a high affinity and still being reversible is the Strep-Tactin XT and Twin-Strep-tag combination. So the Strep-Tactin XT binds with a picomolar affinity to the Twin-Strep-tag, and that makes it suitable for experiments such as BLI. So we show this here as well. So what we did is use the CD45 nanobody that was fused to a Twin-Strep-tag, and we mobilized it on biosensors that were either coated with Strep-Tactin XT or Strep-Tactin. And what you can see is then when you use Strep-Tactin, the nanobody actually very quickly dissociates. So Strep-Tactin is not suitable for BLI experiments. And with XT, you really get this stable immobilization. And that is an example workflow if you use Strep-Tactin XT for BLI. So you have your Strep-Tactin XT probe, which you can purchase ready to use from Gator Bio, or we also offer kits that you can use to coat your own surface. You first equilibrate the probe, then you load your bait protein that should be fused to a Twin-Strep-tag. In the next step, you add your analyte. And then finally, you just have to add guanidine HDL, which will remove the strep-tag protein. And basically the probe is then good to go to be used for another round of measurements. So here I show you some example data. On the left side, you actually find a graph that I took from the product insert that is provided by Gator. So what was done here? They mobilized IL-17A, IL-17F heterodimer, also fused to a Twin-Strep-tag on a Strep-Tactin XT probe, and then different concentration of the ligand IL-17 receptor A was added. And what you can appreciate here is that the baseline is really, really stable. So the protein is really immobilized, and then you nicely see the dose-dependent dissociation and association curves. And here on the right side, we tested the reusability of Strep-Tactin XT biosensors. And again, we immobilized a CD45 nanobody with a Twin-Strep-tag. And we did that for many, many rounds. And even after 30 regeneration cycles, we didn't see a drop in capture level. So the sensors are really stable and can be used many, many times. Yeah, here's now the overview view, so which combination you can now use for BLI experiments. And of course, streptavidin in combination with biotin-related proteins, of course, yes, but you cannot use Strep-Tag2 or Twin-Strep-tag proteins in combination with streptavidin. And also, you cannot regenerate the sensors that easily. For Strep-Tactin XT, you choose a Twin-Strep-tag protein for efficient immobilization, and here you can easily reuse the biosensors. And if you now have a Strep-Tag2 protein and you don't want to change the tag, you have the alternative of going to the Strep-tag antibody, which is suitable for both the Strep-Tag2 and the Twin-Strep-tag. And here you can also regenerate the probes. So now I told you how great the Twin-Strep-tag Strep-Tactin XT interaction is, because it's really strong. You can immobilize a protein, you can use it for purification, especially for low abundant proteins. So now you might be wondering, why does Strep-Tactin still exist, or why do people still choose for the Strep-Tag2? So first of all, this was, of course, the original system, the Strep-Tactin and the Strep-Tac2 combination. And it works perfectly well for all standard proteins. So if you have a protein with high expression levels, you might not necessarily benefit from changing to the high affinity Strep-Tactin XT or from changing the Strep-Tac2 to the Twin-Strep-tag. If you now work with the Strep-Tactin XT and you notice that you cannot elute your protein from it, because this can happen, especially if you have a multimeric protein that not only has just one tag, but several tags, it's possible that you just cannot get it out of the column. And then instead of changing the affinity tag, you can just easily switch to a Strep-Tactin resin, and then you will probably get your protein out of the column. And then on the other side, there are customers who prefer to have this very small tag, or they notice that they're having problems with yield when using Strep-Tactin because the concentration might be low, and then they can just easily switch to the high affinity variant and thereby improve the protein yield while still remaining with a small affinity tag. So here's just an overview of the possible applications. So all combinations can be used for purification, but also for detection, for example, for ELISA or Western blot. And only the Twin-Strep-tag, Strep-Tactin XT combination is additionally suitable also for immobilization. Here I just want to show you what the differences are in the purification cycle between Strep-Tactin and Strep-Tactin XT. Overall, it's pretty similar. So you load your cell lysate containing your protein, you have a washing step, and then the key difference is actually in the elution. Strep-Tactin you have to elute with desdiobiotin, which is a biotin derivative, and Strep-Tactin XT you have to elute with biotin. And you should really keep that in mind, because if you use biotin for Strep-Tactin elution, I mean, you can do that once, it works well, but then you inactivated your columns because biotin binds really strongly to Strep-Tactin, and you can then basically throw your column away. Then the regeneration step is also quite similar. For both versions, you can use 100 millimolar sodium hydroxide for regeneration. For the XT, we actually offer an additional buffer ready to use that contains magnesium chloride, which works equally well. Both versions we have available as agarose beads in suspension format, but also as FPLC columns, gravity flow columns, and also as magnetic beads. And as I briefly mentioned, this tag ligand system is also suitable for detection. So we also have products for ELISA, for example, fluorescent conjugates or microblades coated with Strep-Tactin XT. We have conjugates for Western blot, for example, HRP, and also for flow cytometry. So here we have Strep-Tactin XT, PE, and ABC, which are classical for flow cytometry experiments if you work more cell-based. And of course, for BLI experiments, you can get these probes from Gator Bio, but as I mentioned, we also have a kit that you can use to code your own probes or biosensors. Now I talked a lot about our Strep-Tag products, but since the merger with Cube Biotech, we actually have a very broad portfolio that can support your protein research and especially also AI-guided workflows. So basically, we can support the whole protein journey. We have, for expression, we have different vectors in our portfolio that you can use to express the affinity tags on your protein. We have, for membrane proteins, a stabilization portfolio. We have a broad purification portfolio. And as I mentioned, for the Strep-Tag, also different products for analysis. But what we're really focusing on is this middle part. Especially here for the membrane proteins, we have our native MP technology that consists of many copolymers that you can use to really stabilize your membrane protein in its native lipid environment to preserve the functionality. But we also offer the more classical MSP nanodiscs. And in this purification portfolio, we not only have Strep-tag products, but also for his-tag or Rho1D4 tag. For those of you who don't know Rho1D4, this is a peptide sequence that is captured by an antibody, which is therefore similar to the flag tag, and it works especially well for membrane proteins. And for all tags, we have magnetic beads available, agarose beads, and all in various different formats. And what we especially focusing now on more and more is this automation part, because we also noticed that this becomes more and more relevant, especially in these AI-driven workflows. And those are two products that we recently launched. On the left side here, you can see our Ph.D. plates, which contain pre-plated magnetic beads. So they're in there in a dried format, and those plates are ready to use. We have them for Strep-tag and his-tag. So you don't have to do the plate preparation yourself. You can directly use the plate to perform high throughput purification. And here on the right side, you see our newest product, which are Plate XMP plates, which combine our native MP technology. So really the stabilization of membrane proteins with purification. So you really have all products pre-formatted in one plate. So the copolymers, all the buffers that you need, the magnetic beads, and also the elution buffer. And we have that for Strep-tag, Rho1D4 tag, and flag tag. And using these plates, you can really get a stabilized membrane protein from cell lights in only 2 hours. And we actually also implemented this automation platform in-house, and this was really a game changer for us. So we can really offer many membrane proteins off the shelf. So you can have a look at our catalog. It contains already more than 200 targets and is actually continuously growing. So if you don't want to implement the procedure in your lab, you can just purchase the protein from us and then continue with any assay of your choice. Or if you don't find the target in this list or in this catalog, we also have a custom service where we express the protein, stabilize it, purify it, and also do first analyses and also some assays if needed. And with this, I'm at the end of my presentation. If you have any questions afterwards, feel free to contact me or our customer support. Now I'm looking forward to your questions at the end of this webinar. And thank you very much for your attention.
Hello, everybody. Thank you for joining us. I am Ron Gill, the Innovation Manager at Adaptyv Bio. And today I wanted to tell you about how we are building an automated lab to support digital biology. To introduce digital biology, I will let you hear from Jensen Huang, the CEO of NVIDIA, which is the company with the largest market capitalization in the world these days.
Where do I think the next amazing revolution is going to come? And this is going to be flat out one of the biggest ones ever. And so I told you I was going to tell you, and I'm going to tell you. And there's no question that digital biology is going to be it. For the very first time in our history, in human history, biology has the opportunity to be engineering, not science. When something becomes engineering, not science, it becomes less sporadic and exponentially improving. It can compound on the benefits of the previous years. And every company's contribution, every researcher's contributions compound on each other.
So the idea is great. We will engineer biology. We just need to let computers understand how biology works, and then they can design any protein that we want for therapeutics, for diagnostics, to manufacture goods, etc. However, there is one big problem with reaching this future fast. Computers, they can generate hundreds of designs in minutes, but the lab, the wet lab, takes months to test such a high number of designs, slowing what we call the DBTL cycle. the design, build, test, learn. So every time the computer tries to design a solution, a protein to do something, we have to build it and test it. We feed the results back to the computer. It learns what was good, what wasn't good, and comes with new ideas. And if we could do that fast, the computers can learn from the real wet lab results faster, and we can reach this vision of digital biology faster. And this is what Adaptyv Bio wants to do. We are building a foundry to test proteins. That means an automated data manufacturing line for high throughput testing of proteins to measure their activity, expression, interaction, stability. And this is a service that we sell to our clients, who are mostly people who design protein using computational and AI-based tools. So our users are mainly these AI model designers. Their models comes up with a large number of amino acid sequences for proteins that the model thinks will do the work. Inside Adaptyv Bio, we design and build the DNA that will then be made into the protein, and then the protein will be tested. And these type of work is already being done in general today by legacy CROs, and we plan to do it in a faster, more automated way. So it will also be cheaper, and also we work specifically for computational protein designers, so the results are better packaged both for the software and for people, and I'll show you examples in a minute. So how do we get this high throughput testing? We focus a lot on automation. And our automation is automation on software. Our software follows all the materials. Our software helps us design the experiments. Our software helps us write the codes and the files for the instruments. Most of the pipetting is done using liquid handling instrumentation. We pipette as little as possible by hand. And then the protein readout, their characteristics, is being measured usually, if we can, in automated cells. And automated cells means we have several measurement instruments and a robotic arm and a fridge. So this can run 24 hours 7. We don't need people to feed the instruments only at working time and working hours. And our vision is to reach a self-driving lab. In a self-driving lab, when a customer brings in a project, the computer already understands what are the steps needed, creates all the software and instrumentation files to make it, and the people put like the starting material for it in the right place, and all the automation will run it until data acquisition, getting the results, and pushing it to the customer. And with this view on automation, we are working on a lot of different proteins. So we're currently expressing around 4,000 different design proteins per month. And since we're usually doing several measurements per protein, that means we're creating tens of thousands of data points. And we're hoping to continue to increase this throughput. We expect that even by the end of the year, we'll be at around 10,000 newly designed proteins being tested and expressed every month. And I will show you one side of the automation, and that's the automation related to the Gator workflow. So for customers who are interested to test many different designs against a target that are supposed to be binders, we need to measure the binding affinity. And the Gator tool is the perfect tool for us. And even within the Gator portfolio of sensors, we actually love using the ST-XT probes that you just heard of. And we add to our protein the Twin-Strep-tag in order to have an easy way to load them on the surface with a tag that we can easily add in expression without need for an additional chemical modification. And the workflow starts with us designing a DNA, ordering the DNA, and the DNA arrives. In this case, on the top left, you see in our software tool, how does a plate that of DNA looks like. And the DNA plate goes through several transformations. And if we buy it from an external producer, then it comes lyophilized. We have to reconstitute it. Sometimes we have to dilute it. And then we choose specific DNA construct that will go to expression, which is where we mix them with cell-free based protein expression system to have very fast expression of the different constructs. And then when we have some expression plate and we know that for this specific construct, the customer wants binding data measured, we can tell the software, I want to create a Gator experiments. These are the constructs that I'm going to use. This is the target I'm going to use. And I can have our software generate the BLI plate maps, and also with this BLI plate maps, I can go to the liquid handling instrument. We use a Hamilton liquid handling instrument. The software already created the mapping file that tells the instrument what to do and where source channels on the expression plate should be pipetted into in the BLI plates. And as you can see in this video, in this tool, it has some different heads. On the right, you see 8 independent pipetting channels. They pick up tips, they take liquid from the source plate, and they will go and put this specific amount of liquid in destination plates. And this instrument also can come with other heads, 96 head and 384 head, for higher throughput, depending on what is the mapping that's needed. Then when all the plates are ready, we can add this run into our scheduling software. The scheduling software is the software that controls the robotic arm in the cell. Our internal software also creates the Gator instruction files that take the Gator instrument, which run it should have, and how many probes it should measure, and how many times to dip in each one of the wells. And then we physically load the plate into the back of an automated fridge. You can see on the left, there is some probes plate and some sample plates. And then the other side of an automated fridge, it has a place where the plates will come out. And because this place is fixed, we can easily teach our robotic arm to pick up plates from there. And what will happen is this arm that sits in between 4 different Gator's instruments and this fridge will pick up plates from the fridge and put them on the required instrument. Then the software will tell the instrument, you are ready to go. This is a specific run that you need to do. And the arm will continue and put plates on the other instruments. And when the instrument is finished, the arm will pick up the plates, put it back into the fridge, and bring the plates of the next run. So actually our instruments can run all night and all weekend without people having to put plates into the instruments. This is a little accelerated video that shows the arm in motion over several minutes, putting all these plates. And our software takes automatically the raw data created by the BLI. It does the post-processing of this data and package it on one hand in a package that's easily readable for computers and software for the teaching of AI-based models. And on the other hand, we have different customer views for the people where you can see the ranking of the binding for different constructs, how much it loaded in each case, and we can even like add 3D presentations of the protein-protein interface based on AlphaFolds and other things to help people understand what they're seeing. And all of this, of course, was not done by me. We have a relatively large team. We have 15, around 25, sorry, people today. And half of them are in the biology, running the lab and developing new assays in the lab. And half of them are developing the software and automation that helps the biology people run many more experiments than in a normal lab. With this, I will end. If you have any questions, I'll be happy to answer.
Thank you so much, Dr. Nordlin, Dr. Gill, and Dr. Osborne for your engaging presentation today. We truly appreciate the time and expertise you shared with us. It was a pleasure having you as a part of the webinar. We'll now move on to the final part of today's webinar, the question and answer session. As a reminder, to ask a question, you just need to simply head to the engagement window to the right of the auditorium and click to open the Q&A window. So to kick off this final portion of today's event, the first question I have is for Alexandra. Alexandra, are the plates compatible with Kingfisher devices?
Yeah, that depends a bit on the plate. So the pure HT plates we have available as Kingfisher plates. The plate XMP plates are not compatible with Kingfisher because you actually need a liquid handler that is able to pipette the liquids from one well to the other. But at the end of this month, we actually will launch Kingfisher plates as well, that contain the copolymers in one plate and the magnetic beads in a second plate. And we will provide a protocol also for the Kingfisher system.
That's great to know. Thank you, Alexandra. I've got another question here for Ben. Ben, why would I choose Strep-Tactin XT for a biosensor instead of just a traditional his tag?
That's a great question. This is also one that Alexandra can weigh in on a bit, I'm sure. But the Strep-Tactin biosensor has picomolar binding affinity. That makes it really powerful because it gives a very flat second baseline and it gives a consistency for when you're doing these sort of tight KD measurements. It's also great because for screening, it regenerates well. And so that also gives it a lot of versatility as well.
That's great to know. Thank you, Ben, for that answer. I've got another question here for Ron. Ron, could you walk us through a real example, start to finish? For example, like I upload a design and it ends with actual binding data, highlighting where automation and BLI make things noticeably faster or cleaner.
Thank you for the question. I apologize that the light is working strangely in this place. So actually, we during the second half of my talk, I went through all the steps. So actually, we get a list, normally like a CSV files of amino acid sequences from a client. This is the output of their design. And through the software, we design the DNA that can be expressed using our cell-free expression system. We order this DNA, and when it arrives, we get a plate. As we saw, we put it in our system. Now the plate is existing, in our systems with each construct in a different place. And we have different tasks to be done with it. First, it has to be mixed with the cell-free in order to do expression of the protein. It goes to the incubator, normally overnight. Protein is expressed. We take some of this and we dilute it into, let's say, BLI plates. We also take the target and dilute it into the BLI plates. We load the probes into the probe plate with regeneration buffer. All of these plates go into the fridge that's connected to our automated system, the automation cell that has 4 BLIs. And the software also creates the run file that the BLI needs for each one of the experiments. And then the scheduling software, when it sees that one of the BLI is available, tells the fridge, give out the plate, the arm puts them in the BLI in the right order, and there is a command going to the BLI. Here is the run file with all the instructions, start the run, and the run starts. When the run finishes, all the data is taken from the computers that are all connected to the network into our system. We do the fitting, and usually there is, at the moment, still a human review of the data to be sure that there was no sometimes there's a problem with the liquid handling instrument that in one of its pipetto tips there was an issue and it didn't take liquid, or something happened and there is like a missing point or whatever because some mechanical issue in the end. So we review the data and then we can release it to the client, and the client in their side of the website, they can see the results from the run once it's released.
Thank you, Ron, for that answer. I've got another question here for Ben. Ben, do Strep-Tactin biosensors show a lot of non-specific binding when pulling constructs from cell-free?
That's a great question. So most BLI biosensors are going to show non-specific binding when you put them into a new matrix, right? However, what we typically do when we're doing cell-free expression type experiments is we'll dilute the sample into what we call Q buffer, which has some BSA, a little detergent, and that diluted sample is able to bind, and the non-specific binding is pretty negligible on the Strep-Tactin biosensor. So, and that's even true if there's even moderate amounts of biotin in the solution. It doesn't kick off the Twin-Strep-tag up until you get into very high concentrations of biotin. That's the only time you need to worry about it. So that makes it a very viable tool, and it has a very low limit of detection. So you can bind with very low amount of protein expressed and still get the association and dissociation kinetics data, because we have our XT coding on there that gives you more signal per molecule. So that gives you a lot more signal than you would expect for a tiny amount of protein.
That makes a lot of sense. Thank you, Ben. I've actually got a question here that perhaps both yourself and Ron could answer for me. The question says, for the automated workflow, do you report any protein production data or only the binding data set?
So to answer that from like an instrumentation or what the BLI instrument is capable of, you can look at the initial slope, the initial rate of binding during the ligand loading step from the cell-free, and you can get the concentration of the protein from that loading step. So yes, you do get expression or production data, if you will. But as far as what is used to report to the AI, I'll let Ron answer that question.
Thank you.
So, as Ben mentioned, we collect both the maximum loading signal after our, I think, 180-second loading step, and also the slope. But in parallel, we also most of the time use a method called split GFP, where we have a little piece from the green fluorescent protein that is at the tag of the protein, and you can add in solution, even in a complex solution like the cell-free matrix without a purification, the second part of GFP, and they bind together and they give fluorescence. And with that, you can estimate the amount of expressed protein in the crude format before even you load it on the BLI sensors. And we use that method to see if we had some production issue and none of the proteins expressed or something like that. We don't want to have a full BLI run and then we figured out nothing loaded because there was an issue before. So we use it as part of our QC. And a lot of people, they want to know that something loaded. They don't really are interested in the exact number. There are people who would like to have similar loading for all of their proteins. And there are people who also, like in their models, want to know what proteins are easy to express with E. coli expression. So our cell-free is E. coli based, and what proteins are difficult, because if it's going to be something that will be produced in the future in this system, usually you want a good binder that is also easy to express, because that's a better binder in the total envelope than just one that binds strongly.
That's great. Thank you both for those answers. I've got another question here for Alexandra. Alexandra, since Strep-Tactin XT has an affinity to biotin, can biotin or biotin-related proteins interfere with the BLI assay if I use a Strep-Tactin XT coated biosensor?
Yeah, I think Ben kind of mentioned this in his answer also already a bit. So yes, there is a bit of affinity to biotin, but the affinity of Twin-Strep-tag is much stronger. So it basically does not have that much of an effect. And I think Gator also showed that in an experiment that a biotin concentration that's around a normal level in a cell culture media usually does not interfere with the assay. If you have higher biotin concentrations, you can always add avidin, because avidin really specifically blocks only the biotin and also biotin-related proteins and not the Twin-Strep-tag proteins.
That's great. Thank you, Alexandra. The next question here is for Ron. Ron, when looking at the BLI outputs to train the AI, which numbers do you use to refine the AI model? Is it just KD?
So I think it depends on what is the model, and people have like models with different, let's call it, outcomes that they want to get out of it. A lot of people, their KD is enough, even though we get K on and K off. But some people, for example, for in vitro diagnostics, you're actually more interested to have slow K off rather than the strongest KD possible. And people who want to like develop models to make better binders for in vitro diagnostics, they will want models that can predict also what is the best K off and not only what is the best KD. And in that case, they will want this information to feed their models in order to improve its predictions. So it all depends on what the people who have the models want the model to predict.
That's great to know. Thank you, Ron. The next question here is for Alexandra. Can I use an AviTag / biotinylation and a Twin-Strep-tag on the same protein?
Well, we usually don't recommend to use both tags on one protein because you cannot really 100% exclude interference. But if you now have a strep tag for purification and then you biotinylate the avi tag afterwards to immobilize your protein on a streptavidin chip, I wouldn't expect it to be that much of a problem because the binding of the biotinylated protein is very strong. If you have a twin strep tag, you basically could save the biotinylation step because the twin strep tag can do the same as the avi tag can do.
That makes sense. Thank you, Alexandra. The next question I have here is for Ben. Ben, what is the lowest concentration that Strep-Tactin can detect on BLI?
That is a great question. You can get down into the low micrograms per mil, even lower. You can get into the nanograms per mil range. The lower your concentration is, the longer the loading step needs to be in order to get enough protein to really know that you've got something there. Because BLI measures things in real time. So we're actually, you'll spend, typically with a cell-free expression type experiment, you'll do 1 or 2 minutes of the loading step from the cell-free, right?
But if you know that you have one that expresses poorly or that is low, you can extend that time, and you can extend it live. You can as you're running the experiment, you can add a bit more time, a few more seconds to the experiment to know that you've got enough for the next experiment. Typically, however, if you have something that is that low among a bunch of other candidates, then you know to exclude that because it's a poor expressor.
That's great to know, thank you. The next question I have here is for Ron. Ron, from your perspective, what are the biggest pain points in a normal wet lab validation workflow, and which of those does an automated setup pretty much wipe out?
So, when people are working in the lab, okay, even people who are very good at what they do, sometimes they make mistake. They pipette into, let's say, the wrong well. They thought that we needed target 1, but we actually needed target 2. And because we have in a high throughput measurement lab, we have so many steps for so many projects, so much happening in parallel, the chance that there will be a human mistake somewhere along the process is high every single day just because we are running so many projects. And therefore, we need instruments that they go every time to the same spot. They take every time from the same well to the other well. So it's not that there are no issues, okay? Sometimes we programmed maybe the instrument wrong, but then we find it out and we fix it for all of the next experiments. And once in a while, I don't know, one of the tip boxes was not arranged well and there is an issue, but there is not like these issues that sometimes people will forget something along the way that is unfortunately will happen when you have like 20 different steps and 50 different projects run by 10 different people in the lab every day. And that's where we hope automation will help us. Some people think that automation will make things much faster than humans. Actually, our pipetting robot is slower than a human, but it does much less mistake and it can work all night without asking for pizza. That's my answer. Thank you.
Thank you for that answer. I've got a question here, but I'm not sure who to direct it to, so perhaps you can just jump in. Someone is asking how to balance the protein capture level for different expression levels.
That's a good question, yeah. And this is the Ron and I kind of question. So it was mentioned a little bit before that you're going to, when you do a cell-free expression with a dozen or 100 candidates, you don't actually know how much protein's in those wells. You don't. So what you do is you capture as much as binds, and then you do a single point measurement with that and get an idea of the concentration based on that binding response. And then you get a kinetics measurement as you interact that in a new well with the target protein. That target protein binding is generally speaking, it doesn't matter how much protein is on the probe. You're going to see how much binding on and how much off you get. Yes, we can get into the kinetics, right? And yes, we can optimize the experiment. But for a screening approach, all you need to know is yes, no, is there something there? And does it stay on? What's the K off? Get a general idea of the KD. So that's how you typically will approach this. You say, let's set it for 2 minutes, capture time. You bind as much as is there, and you see how much there is at the end, and then you optimize. Because again, when you're doing cell-free, you're trying to get answers quickly, and you accept that you don't know some things when you run that first experiment. But when you're using a Strep-Tactin biosensor, which can regenerate numerous times, the cost per assay is under $1. So it's very approachable in terms of doing a preliminary experiment to get started, and then you're able to refine it with those same biosensors later.
That's great, thank you, Ben. I don't know if Ron, you wanted to jump in on that one?
I think that Ben gave a very good answer. I don't have much more to
That's
add
okay.
to that.
No problem, thank you. And I've got another question here for you, Ben, as well. What's the throughput per week for a Gator BLI instrument?
Actually, Ron is also very well versed in
Oh,
that.
okay. Well, yes.
We're talking on the order of thousands, right? And you said you're running
Yeah.
4,000 a month right now, Ron?
Yes, we have like 4 BLIs, but we have also other instruments, etc. First of all, it's important to say that BLI has different instruments with different throughput for a wide range of user requirements. So if the instrument has only 8 channels in parallel, the throughput will be less than if the instruments have 32 channels in parallel. And then normally, with the Pro, for example, which is the larger number of channel instruments, if we do 5 concentrations, we can run around 96 different proteins in about 6 hours. So we're talking about 300 full KD measurements in a day, something like that.
Exactly. Yeah, and it depends on the kind of experiment you're running, right? Ron was explaining if you're doing like a proper kinetic characterization of your molecules, 300 a day is a very reasonable and very high throughput number, frankly. But if you're just doing basic screening type experiments, single point, just doing quick measurements, you're often you can squeeze in far more. You can do far more because the experiments are faster. You're looking for quick yes/no data instead of a controlled specific experiment where you're trying to really nail down a real KD value.
That's great. Thank you both for that answer. But I'm afraid that is all we have time for today. I'd like to give a huge thank you to our amazing guest speakers, Dr. Nordlin, Dr. Gill, and Dr. Osborne, for sharing your knowledge and expertise. And of course, thank you to all of you who joined us today. We hope you found the discussion insightful and that you walk away with new ideas and perspectives. If we weren't able to get to your question today, don't worry, we'll be following up with the answers after the webinar. And if you have any additional questions after the session, please feel free to reach out to me at the email address editor@selectscience.net. Certificates of attendance will be automatically emailed to all live attendees a few days after the event. For those watching on demand, a certificate can be requested by emailing us at the same email address. The on-demand recording will be available in just a few days, allowing you to revisit the webinar or share it with colleagues or friends. Once again, thank you to everyone for attending. Stay tuned for updates on future webinars, and we hope to see you again soon. Take care and have a wonderful day ahead.